Good morning, everyone. Welcome back. My name is Bruna Florencio. I'm a graduate student at the Federal University of Goiás. It's my pleasure to welcome you to this section that will be presented by João. He's a forest engineer and quantitative geneticist at Clabin, working in the forestry breeding program. He holds a master in forest science and has about 7 years of experience in plant breeding, quantitative genetics, and applied predictive modeling. His works covers multi-environmental data analysis, genomic prediction, decision support tools, and integrating environmental information to improve selection and clonal deployment. He is currently a PhD candidate at the Federal University of Paraná in plant production, and is affiliated with the graduate program in agronomy. Before we begin, I would like to remind everyone that we'll have the question and answer section at the end of the lecture. So please feel free to submit your questions and comments in the chat through the presentation. So it's a pleasure to have you here with us. You may start your presentation.
Thank you, Bruna, for the introduction. So, hi everyone. Today, I'll be talking about environomics as a tool to guide clonal deployments in Eucalyptus. But before getting into the technical part, I'll briefly introduce Clabin and the company context behind this work. So, Clabin is a Brazilian forest-based company and the leader in Brazil in the manufacture of coated board, corrugated boxes, and industrial bags. The company also supplies the market with short fiber, long fiber, and fluff pulp solutions. And today, Clabin operates 24 industrial plants, most of them in Brazil, and has commercial offices across several Brazilian states, as well as international presence in North America and Europe. And an operation of this size requires a large workforce. So Clabin has more than 8,000 direct employees across forestry, industry, logistics, commercial, and all the technical areas. So this slide, it gives like an overview of Clabin's geographic footprint. So the company, like I said, it operates across several Brazilian states and also has an operation in Argentina. So focusing on forestry a little more, which is a central part of Clabin's business. So the company, it manages around 908,000 hectares of forest area, mainly in southern and southeastern Brazil with both eucalyptus and pine plantations. And around 41% of this total area is preserved native forest. So the differential for Clabin is working with both eucalyptus and pine, and this gives the company a lot of flexibility in terms of fiber supply and product development. So Pinus, it provides the long fiber, while eucalyptus provides the short fiber. And these fiber types, they have different properties, and they allow Clabin to like serve different industrial applications. So long story short, Clabin is an integrated forest-based company where the value chain, it starts in forestry, moves through pulp and paper, and reaches different packaging solutions. So this integration, it connects directly the forest base to the industrial products and the final market. But like I mentioned, Clabin works with pine and eucalyptus, but today I'll focus on eucalyptus. So why eucalyptus? So eucalyptus is a very important genus for planet forest because it combines three characteristics that are like extremely important and valuable for breeding and industrial forestry. So the first one, eucalyptus has a high genetic diversity. So there are more than 700 described species with like extensive genetic variation both within and between those species. And this, it gives breeding programs a very broad genetic base to work with. So we can use eucalyptus as pure species, but there's also like strong potential for hybridization. And because the clonal propagation is relatively easy, once a superior genotype is identified, we can fix and deploy those genetic gains over large operational areas. And also, eucalyptus has a fast growth, especially in tropical and subtropical regions like Brazil. So, rotation cycles here are commonly around 5 to 7 years, which is very short, at least for a forest crop. And, and third, eucalyptus has a major role in the Brazilian economy. So, Brazil has more than 10 million hectares of planted forest, most of them with eucalyptus, and the country is also one of the main global players in pulp productions and exports. But even with the fast growth and all, developing and validating a new elite clone is not something like trivial, it's not immediate. So this slide it shows like a simplified view of a typical eucalyptus breeding cycle, and of course each company may follow slightly different strategies, but the general logic is usually similar. So the process it starts with crossings where controlled pollination is used to generate this new genetic combination. So in other words, we are like generating this raw genetic variability that will be evaluated later, and then we move it to the progeny trials we where we estimate the genetic merit and select superior individuals. And after that, those selected individuals they move into clonal trials where we evaluate clonal performance more directly, and we start confirming whether the genetic gain that we observed earlier is actually expressed when the material is clonally propagated. And then finally we have the pilot planting, which is like closer to an operational reality. The clones are competing with itself, and the objective here is like a final validation of commercial planting before the large scale deployment. And of course, with tools like genomic selection, we can shorten this timeline, but genomics is not the focus of today's talk, so I won't go in that discussion here. And so the main point that I want to emphasize with this is that clonal deployment is the result of like a long and expensive decision process. So by the time a clone reaches the commercial planting, we really need to understand not only its average performance, but how reliable that performance is across environments. And that's why testing across multiple environments is such a big deal. It's such an important part of the breeding process. So, in a simple way, when we evaluate genotypes across multiple sites, we can separate two important sources of informations. And one is the genotype effect, which like reflects an overall genetic merit of the material, and the other is the G by E effect, which reflects how that genotype responds differently depending on the environment. So this is very important because different breeding and deployment objectives can lead to different choices. So sometimes we want a genotype that performs well overall and is stable across many environments, but other times we may accept a more specifically adapted genotype if like it's performed extremely well in a particular site or region. So multi-environment trials are essential because they allow us to evaluate both. And over time, several tools have been developed to analyze this type of data and to help breeders to understand and interpret G by E. And here we have a few of them. So one classical approach is the AMI, AMMI. So this approach became widely used because it helps like to decompose G by E patterns and visualize which genotypes and environments contribute most to the interaction. And another very popular tool is the GGE by plot, a very useful tool to like visualize genotype performance, mega environments, and which one where patterns. And we also have some BLUP-based indices like this one, like the MHPRVG, which combines metrics of productivity, stability, and adaptability to like support genotype selection. And we also have the weighted average of absolute scores or WAS, which is a more recent approach, which is kind of kind of a mix, kind of a bridge between block-based indices and the ME decomposition. And one nice thing, especially in the Metam package, is that we can generate these intuitive quadrant plots that you can see on the right, combining mean performance and stability, making it easier to like classify genotypes and environments in a very systematic way. So with all of that being said, today I want to try to answer a simple question. So is G by E a constant problem? Well, if you start to think of it, it is a fact that it has an impact on genetic gain. So when G by E is present, the same genotype does not express the same phenotype everywhere. And I mean, that sounds really obvious, but the consequences are not trivial. Because it directly reduces the heritability and consequently the selection accuracy for the trait that you're trying to select. And second, the deployment complications. So in a scenario with strong G by E, it is very difficult to extrapolate results from trials to untested sites. And deployment is done mostly in untested areas. So, like, we can't test every pixel of the landscape, so we need to generalize a little. And in forestry in particular, this type of decision can become a little problematic since like a wrong decision can be easily replicated across several hectares of land. And well, you're like stuck with that decision for at least 5 to 7 years, which is the average eucalyptus cycle in Brazil. And there's also some model limitations. So those tools that I showed you on the previous slide, they are very nice, they are very powerful, but they're limited. They're limited to describe and interpret G by E because they treat the environment as a factor level, not as something measurable or quantifiable. So we end up in almost a paradox where we can quantify and visualize G by E very well, but the moment we need to deploy into untested space, which is the real world problem, the classic toolbox is often not sufficient by itself. So asking again, is G by E a constant problem? Well, like everything in the world, it depends. So the answer is yes and no. But if we change our way of thinking, if we change our mentality, and if we accept that genotypes have different response patterns, then G by E can become structured information. And in that sense, G by E can become an opportunity because it literally tells us which clone or which genotype is best where and under which environmental profile. But how do we explore it? How do we explore GBI? How we move it from being a problem to become like useful information for deployment? It's simple, yet it's not that simple, but by predicting it, by predicting GBI. All right, so if we agree with the conclusion from the paradox slide, the next question is how did we get to the point where predicting GBI is even feasible at all? So this is something that did not appear out of nowhere. It was like a gradual shift in plant breeding thinking. And this timelines, it tries to highlight some key milestones and researchers behind that process. So first, when going back to 2006, this is when the idea of combining environmental information with geographical information systems started to gain some traction as a practical breeding tool. So the site, it stopped being just some random name on a spreadsheet and started becoming a location in space that can be like mapped and compared and used to support recommendations beyond the trial itself. And then there's a big conceptual jump in 2014, which is, in my opinion, probably the most important milestone. Haslott and Harkin, they formalized the idea of reaction norms driven by environmental covariates. So basically, the idea of treating genotype as a function of measurable environmental descriptors, not only as some categorical and environmental label. And the idea is to allow that genotypes can have different sensitivities, different slopes, and that is already a form of predictive G by E. And between roughly 2005 and 15 and 2019, several studies they start to demonstrate that this isn't just some kind of elegant idea, some nice concept. It actually works. And also, it was in 2019 when the term environomics was first introduced. And by 2021, environomics became more consolidated as a breeding framework. So this is where predicting GBI stopped being like a niche methodology and became a coherent pipeline. And I also would like to highlight that a lot of the researchers shown here are Brazilian, and as a Brazilian myself, that makes me very happy. Okay, so first to understand environomics and how it actually works, it's important to understand some new terminologies that are borrowed from genomics, and it represents like important processes inside the whole environments pipeline. So starting with envirotyping. Envirotyping is very simple. It's just the process of measuring and characterizing the environment in a systematic way. So when we are like gathering information, environmental information, or even management information of a specific piece of land, we are envirotyping it. And then we have the environomic markers or EMs. So the analogy to genomics is intentional. Just like SNPs are markers used to model the genetic variation, the environomic markers are these environmental features used to model the environmental variation and its interaction with genotypes. So for us, they are the modeling units. I'll talk a little more about environmental markers on the next slides, but for now, just think of them as modeling units. And we also have the envirom, which is the combination of these environmental markers that describes a location. So if you think of the genome as a combination of genetic markers describing an individual, the environment is basically the same thing. It's a set of environmental markers that describes a location in space or the growing conditions for a pixel or a site. And then also the term envirotype, it refers to an environmental profile at a site or pixel. So each value or feature that an environmental marker can adopt inside a specific piece of land is an envirotype. Okay, so talking a little more on environmental markers, I want to highlight that similar to molecular markers, we also have some premises that are like good to be considered when choosing an environmental markers. And the first one, the linearity. So I'm not saying that biology is linear because we know that's not the case. What I'm saying is that for a given marker, we want a relationship that can be like reasonably approximated by a simple function, which is often linear. And the site potential, actually, the value of a mark should ideally reflect something about how favorable that site is for expressing the trait. So when the marker is high, we expect like higher potential for growth or for whatever trait you're modeling. And also there's the heterogeneous favorability, which is probably the most important one. Basically, it's the core of GBI. So genotypes, they do respond differently to the same environmental marker. If all genotypes responded the same way, there would be no interaction, right? So for an environnomic marker to be meaningful for GBI, it has to allow these different responses. So in a way, this is similar to polymorphism in genomics. So a DNA marker is only informative because it varies across individuals. So the idea is pretty much the same. And there's also the EC genotype dependence. So not every environmental covariate matters for every genotype in the same way. A covariate can be like highly informative for one genotype or for a group of genotypes and almost irrelevant for another. Okay, so how do we actually build and choose these markers from the environmental covariates? Well, we have a few options. The first one is we can use the raw covariate itself as a marker. So this is the simplest and most interpretable approach. So if you have a covariate for temperature, rainfall, soil, clay content, or maybe some remote sensing index, you can include that directly in the model. So the limitation is that it assumes that there is a reasonably simple relationship with the phenotype, and you can struggle a little with like strong non-linearities or maybe highly correlated covariates. And we can also transform those covariates. So starting from the same raw variables that I mentioned, we can apply like some transformations like logs, polynomials, or maybe splines to try to capture part of that nonlinear response. And we can also derive some ecophysiological variables from these covariates. So the idea here is like we want to translate the environmental data into an index that is more related to some plant process, things such as growing degree days or maybe some metric of water stress. The advantage is that these markers, they carry more biological meaning. And we can also combine these markers into kernel matrices. So instead of modeling each marker, we can build environment by environment similarity matrices, similar to like build a relationship matrix in a GBLUP approach. So the idea is pretty much the same. And we can also work with engineered environomic markers, which are synthetic markers. So the idea is to use some machine learning approach to learn the complex relationship between environment and phenotype. So one downside of this is that these markers, they lose their direct interpretability, but on the good side, it's fairly easy to use them as predictors to GBI. Okay, so after talking about what environomics is and what environomic markers are, how does environomics work in practice? So on the left side, we have the input side of the pipeline, the data set building. And the very first operational step is the definition of what we call a target population of environments, or TPE, which is basically our area of interest. So once we define this area, once we define the TPE, we start to think of it as a continuous landscape that we want to make decisions on. So the idea is that we divide that landscape into a grid of pixels. And some of these pixels, they contain phenotypic information because they include field trials or maybe inventory plots or any real observation of what was planted there and how much it produced. And other pixels, they have no phenotype at all. And also, if genotypic information is also available, like DNA markers, you can incorporate that as well. But the core idea of environomics does not depend on genomics. And now moving to the modeling step. So the idea, it's really straightforward. So we use the pixels where phenotype is available as the training set, and using that information, we fit a predictive model that learns how genotype performance changes across the different environmental conditions. So in other words, the model learns G by E. And once the model is trained and properly validated, we can apply it to the rest of the TPE and predict the performance of all genotypes in every pixel. Okay, so now I'll switch from concepts to a real operational case. So in this work, we use an environmic framework to like predict productivity across ATPE and translate those predictions into clonal deployment decisions. So starting with the setup of the study, so talking about the data set, in this study, we worked with 15 eucalyptus clones from 5 different species that were evaluated using around 5,000 operational inventory plots distributed across 3 Brazilian states. And our response variable was mean annual increment standardized at 7 years, this MAI7, which is just a common measurement of productivity in forestry. Just know that. And then, like I said, the very first operational step is the definition of the TPE. And we did that by connecting the outermost plots and adding a buffer. And then talking about the spatial scale, here we use then 1 kilometer by 1 kilometer grid across the TPE, resulting in approximately 208,000 pixels, a lot of pixels. And in this study, we used those engineered environmental markers that I talked about, and they were built using almost 4,000 environmental covariates that were envirotyped across the TPE. So we built those synthetic markers using random forests to transform the raw covariates to 10,000 engineered environmental markers that carry some predictive signal for the trait of interest, while aggregating those nonlinear responses. And then we validate and organize these markers. We can use like random forest out of bag validation to identify and remove poorly performing markers. And then because the remaining markers, they still contain like considerable redundancy, we group them into 8 clusters using hierarchical clustering. Remember that information because it's important. And to validate the model, we use a leave one region out cross validation. So we clustered the data set into 20 spatial regions based on geographic proximity, as you can see on the map on the right. So at each fold, one region is removed, the model is training on the remaining regions, and then we predict that one region. This process is repeated 20 times until every region is predicted, a standard cross-validation approach. And we also built some controls and some benchmarks to interpret and to compare the environmental model and to interpret what the model was actually learning. So the first control is just a baseline G by E model, which is just a simple model using genotype, region, and genotype by region as random effects, but without any environmental covariates or environmental markers at all. And the other benchmark is something that we call a false genotype prediction test. So the idea behind this is to check whether predictions are like truly capturing genotype specific responses rather than mostly some kind of environmental pattern generally. So in each validation fold, each pixel was predicted using the reaction arm of a random genotype that is not present there. Then that prediction is compared with a genotype that is actually present there. So if the model were basically learning some kind of environment quality, this false genotype scenario would perform similarly to the real environmental model. But if the real environmental model is indeed capturing G by E, the performance in this scenario should drop. And then finally, the prediction. So we use mixed effects modeling in an ensemble-based framework for the validation too, of course, I forgot to mention. But so the model, it contained like a fixed intercept and some random genetic terms that include genotype-specific reaction norms along the environomic marker. So in essence, that means that each genotype has its own baseline and its own sensitivity to the markers. And then these predictions are repeated 1,000 times. So in each run, one marker was sampled from each cluster. Remember that I said that we clustered the markers, creating a different set of environmental predictors. In essence, a different model. So then at the end, those predictions are average, a standard ensemble framework. Okay, so here I want to present our cross-validation results. So the baseline G by E model is what we get without any environmental covariates or markers. So we get a correlation of 0.39 and a root mean squared error of 8.73. So in essence, we have like limited abilities to preserve genotype rankings, and the prediction error is relatively high. And in the middle, we have the environomic model that was built with those synthetic markers, those engineered environomic markers. And we can see that this is a completely different picture. So correlation jumps to 0.92 and root mean squared error drops to 3.09, a much, much better result. And now this third box is the control that I mentioned, the false genotype prediction test. So here, just to remember, we intentionally predicted each pixel using a genotype that is not present there and compare it to the genotype that is actually present there. And that result is actually very informative because it confirms two things. The first one, it confirms that the environment alone, it carries predictive power, which is expected. But more importantly, the gap between the real environomic model and the false genotype scenario is an indirect evidence that the model is indeed capturing genotype specific responses, that the model is indeed capturing G by E. Okay, so my slide, it's stuck.
Yes, it's not passing the slide.
Oh, why? Okay,
Now it's okay.
okay. Now, okay. Probably because the animation, I think. I don't know. So you're
That's
seeing
all.
it correctly, right?
Yes, perfectly.
Okay, okay.
You may continue.
Thank you. So what you're seeing here is a spatial prediction across the entire TPE. So each color of this map on the left, it corresponds to the top ranked clone per pixel based only on predicted productivity. So the model, it obviously does not only provide the winner. So for every pixel, we can obtain predicted productivity for every clone that we evaluate, which is gold for decision making. So, and now one thing that's immediately clear here is that out of the 15 clones, recommendations they concentrate in a small portfolio. So these top 5 clones, they cover 93% of the TPE, and clone G5, the green one, it dominates more than half the area. And I want to talk a little more about these 5 clones because this information is important for the next step that we took. So we have some tropical clones, the G5, the G14, and G8. They are hybrids between Eucalyptus grandis and Eucalyptus urophylla, which are two tropical eucalyptus species. And then we have an intermediate clone, G7, which is an Eucalyptus saligna, and a subtropical clone that is from a species more adapted to cold regions, Eucalyptus duni, the G2, the blue one. And this matters because our area, our TPE, is located in a climatic transition area that include both warm and colder areas. And a lot of these cold areas are very frost prone. And I don't know if you know that, but it takes only one severe frost to like strongly damage entire plantations of a susceptible genotype, bringing massive financial loss. So that's why the next step that we took was not to replace this map, but to refine it by integrating a frost risk layer. So, and first of all, I need to say that the environomic model is not absolute, so it needs to be interpreted with caution, with some operational risk in mind. And the breeder's expertise is extremely important. So there are biotic and abiotic risks that are very difficult to fully capture or quantify in a predictive model. And because of that, the best decisions they come from combining data-driven predictions with field experience and operational knowledge. And like I said, in Clabin's case, frost is extremely important. So while the previous map it showed the productivity-based recommendation, it does not yet accounted for this type of risks. And I mean, it's not the model's fault that we don't have many observations of susceptible genotypes in highly frost-prone areas, because to be honest, it would be a little stupid to plant those genotypes there because they'll die. So With that in mind, we refine the environmental predictions by incorporating this clone-specific susceptibilities to frost, using this minimum temperature classification map on the right. And this was implemented as like a second step in the deployment process. So instead of replacing the original predictions, we were trying to adjust them to account for an operational risk. And this logic, it can be extended to other types of risk as well. Uh, Almost any kind of risk that are relevant for a given genotype or region, such as the occurrence of a specific disease or pest outbreak or maybe drought. So what you see here is the results of that combination. So looking at the final map, the one on the right, we can see some important shifts in the colder regions where the G2, the Eucalyptus duni clone, it increases substantially in recommendation because it is more tolerant to frost than tropical materials. And we can also see a new clone, G12, appearing in these harsh frost-prone areas where even G2 may not be the safest option. So this is a clone of Eucalyptus bentami, uh, Eucalyptus bentami is a species well known for being very frost resistant. Compared to the other options, it may be not the best performer in productivity alone, and that's why it didn't appear on the first map. But out of the 15 clones, it's like the only species capable of surviving in these harsh environments. And finally, if we compare the recommended clones with the ones currently planted, we can observe a positive distribution of gains, with an average expected increase of about 13%, which is very significant. So to like wrap everything up, this slide it tries to summarize the full environmental pipeline that we applied. So we start, like I said, the very first operational step. We start by defining the TP, the area, and collecting data. So phenotypes from inventory plots and a large set of environmental covariates across every pixel of the landscape. And then these covariates are then processed with productivity data to generate the synthetic markers, the engineered environomic markers, capturing those complex environmental patterns. And then using these markers, we can fit a predictive mixed effects model with spatial cross validation, allowing us to learn genotype specific responses and predict performance across the entire TPE, predict G by E. And from that, we generate a productive based recommendation, identifying like the best clone in each pixel. But as we discussed, prediction alone may not be enough. So in a second step, we integrate an operational risk, in this case, frost susceptibility, to adjust recommendations and make them more robust. And the final result is a deployment map that combines prediction and risk. Okay, so beyond deployment maps, Enviromics also provides some interesting and useful results. So the environmental potential, for example, this is the predicted productivity at each pixel using only the markers. So in practical terms, it gives like a map of a baseline site quality across the TPE. We can also estimate the genetic variance. So the idea is like here we look to how strongly the predicted performance differs among genotypes at each pixel. So here the map on the left it shows the genetic variance. So in our results, some regions at the edges of the TPE showed like a higher genetic differentiation among the predicting clones. So in other words, a higher discriminant power. And this pattern is also consistent with what we observe in field evaluation in those areas, even though those specific results are not shown here. And now looking at the map on the right, the environmental potential one, where each pixel represents the baseline, site quality and all. So what is interesting about these two maps is that we can see like a spatial contrast between these two areas. So areas with lower environmental potential, they tend to show a higher genetic differentiation among genotypes. So the best environments are not always the most informative for selection. Some lower potential or maybe more challenging areas can be exactly where clone differences become more visible. And we also have the breeding zones, those areas that minimize G by E. So the idea behind this is not to cluster environments by climate alone. The idea is to cluster the area, cluster the landscape by how genotypes behave and create those zones where G by E is minimized and rankings tends to be more consistent. So you can see here that the within zone correlations are high, while between zone correlations tend to be lower and can even be negative. And I don't know if you noticed it, but that question mark in the title is intentional because someone can argue that these are not breeding zones in the strict sense, and for two reasons actually. The first one is the clustering is based only on operational data rather than like a network of breeding trials. And the second one is like we work with only 15 eucalyptus clones, which may be limited for fully characterizing a TPE of this size. So maybe a more precise term here would be deployment zones, but the main point it remains the same. The idea is to capture structured G by E patterns and help them, like help organize decisions across a landscape. So to finish, I want to highlight where I believe Enviromics is heading as a tool for predictive breeding and deployment. So the first one, Enviromics as a deployment tool, I talked a lot about that because I truly believe that Enviromics has the potential to become like a standard approach for genotype deployment. And also one thing that I talked about, decision making needs to be synergistic. I mean, models are powerful. No one here is saying otherwise, but they should not replace expertise. They should, complement it. And there's also multiomics integration. So there are already some strong examples showing that combining environomics with genomics and phenomics can improve predictions. And I mean, that's definitely the future. And finally, integration with breeding trials. So connecting operational data with trial data can be very powerful because you can improve prediction of new genotypes, and you can also help to bridge the gap between performance on on breeding trials and performance after deployment. So overall, the path forward is clear, like more integrated, data driven, synergistic, and spatially decision making systems. And this not only for forestry, but for any crop at all. So that's all I had for today, folks. Thank you.
Thank you so much. It was a pleasure to listen to your presentation. We'll now move to our question and answer section. Just a moment. I'm going to
Okay.
read it. The first question is from Camila Barros. Do you see environmics-based models eventually reducing the need for extensive multi-environmental field trials, or they mainly serve as a complementary tool?
If I see that environomics can reduce breeding trials, is that right? Yeah.
Yes, that's it.
Okay. Oh, if we apply it, I mean, that's not the case with this study because we focus on inventory plots. But if we apply it at breeding trials, yeah, we can like predict genotype performance in locations that there was never a breeding trial. So yeah, we can use it that way, and it's a very powerful way to use it.
Okay, thank you for the answer.
Okay.
The next one is from Larissa Reis. In your experience working with Eucalyptus, are environomics approaches predictive models with environomics covariates already being used in operational decision making in breeding programs?
If we are applying this operationally, yeah.
Yes.
Oh, this study was like a pilot study to see if the whole pipeline, how it would behave with our data. but but yeah, we are trying to use that operationally in a second model fit, that we are including more data, um, more clones and all. We are also focusing on a small area, focusing only on deployment. So yeah, we are like almost there, if I had to say. Yeah.
Okay, thank you again. And the next one, it's more a comment than a question. Um, João Batista, that's a professor here at UFG, he think about the patriotism, like being proud of being Brazilian on this. So he's really
Yeah,
proud
yeah,
of
yeah.
it.
That's nice. That's nice.
And he has another question.
Okay.
Wait just a second. They are asking if you have the GitHub account or database that you can share with Enviromic analytical pipeline.
Unfortunately, no. Sorry.
Let me see. So that's it about the questions.
Okay.
Thank you very much one more time. It's been a pleasure being here with you today.
My pleasure. Yeah. That's a nice.
And so, just some information. We are having a lunch break right now, and we're going to be back at 2 p.m. at Brasilia time. The link will be on the chat again, and we'll be with Salvador Gezan when we are back. So that's it. Bye bye. Thank you one more time.